Hello viewers, welcome to Zoological Society of Assam. Today I am going to present before you the replication of viruses. Let me introduce first. I am Dr. Shebara, HOD and Associate Professor, Department of Zoology, Guru Charan College, Silchar. Learning outcome of this presentation are the students will get the general idea about different types of cycles of the viruses. Secondly, the students will be able to distinguish the inner mechanism of the viral cycles. And thirdly, they will get the concrete idea about how viruses act inside a host cell. To begin with the topic, let us talk about the viruses. Viruses are small, submicroscopic, intracellular obligatory parasites which contain either DNA or RNA genome surrounded by a protective virus-coded protein coat. A complete virus particle is called a virion. The main function of the virion is to deliver its DNA or RNA genome into the host cell so that the genome can be expressed by the host cell. Now the replication of virus is very complicated process. Viruses never reproduce by division as the other cells do. They are replicated by a process in which components of virus are produced separately and are assembled into intact virions. For replication of virus, host is necessary and viruses are host specific. That means each virus has its own specific host. A host may be a bacteria, a plant, or an animal. Replication of viruses are studied for the first time by experimenting on bacteriophage of the T series like T2, T4, T6, etc. There are two types of life cycle commonly seen in viruses. These are the lytic cycle and the lysogenic cycle. The lytic cycle involves the reproduction of viruses using a host cell to manufacture more viruses. The viruses then burst out of the cell. The lysogenic cycle involves the incorporation of the viral genome into the host cell genome, infecting it from within. In the lytic cycle, sometimes referred to as virulent infection, the infecting phage ultimately kill the host cell to produce many of their own progeny. Immediately following injection into host cell, the phage genome synthesizes early proteins that break down the host DNA, allowing the phage to take control of the cellular machinery. The phage then uses the host cell to synthesize the remaining proteins required to build new phage particles. Ultimately, the heads and sheaths are assembled separately and the new genetic material, like new daughter phage particles, are constructed. During this process, the host cells gradually become weakened by phage enzymes and eventually burst, releasing on average of 100 to 200 new phage progeny into the surrounding environment. Certain bacteriophages such as P1 and lambda phages have entirely different pattern of life cycle than the virulent phage. This pattern is called lysogeny and is characterized by delayed lysis after phage infection. A virus with this capacity is called temperate virus and the infected host cell is said to be lysogenic because dormant virus may at any time become active and begin directing the synthesis of new virus particles. In the lysogenic cycle, what happens? Sometimes it is referred to as temperate or non-virulent infection. It does not kill the host cell, instead using it as a refuge where it exists in a dormant state. Following the injection of the phage DNA into the host cell, it integrates itself into host genome where it is then termed a prophage. The prophage genome is then replicated passively along with the host genome as the host cell divides, and then transition occurs from lysogenic to lytic cycle. Now let us talk about the key steps in the viral replication cycle. The first one is attachment, second is penetration or entry, third is uncoating, fourth is genome replication, fifth is assembly, and sixth is maturation and ultimately release. The first event in replication of viruses is the attachment. A virus is host specific and enters into the specific host or target cell. This event is electrostatic and does not require any cellular or metabolic energy. And virus exhibits cellular tropism. Also, virus has host range which may be narrow or broad. For example, HIV virus which has broad range. A virus uses receptors and antireceptors for attachment and entry into the host cell. Next, cellular receptors and antireceptors are mostly proteins, but sometimes they may be glycoprotein, carbohydrates, or lipids. The presence of virus-specific receptor is necessary in all the cases. For example, HIV has CD4 receptors, rabies has acetylcholine phospholipids. Now, second step is the penetration or entry. Penetration is an energy-dependent process. Virus may be penetrated into the host cell by endocytosis or translocation. The third step in viral replication is uncoating. It refers to the removal of degradation of capsid, thereby releasing the genome into host cell. The virus genome is transported to the site where transcription or replication can begin, and in some cases there is no degradation of capsid, as capsid proteins play a role in viral transcription and replication. Fourth step is genome replication. Viral genetic material or genome is multiplied within the host. Simultaneously, viral structural proteins like capsids are synthesized. The type of genetic material varies from virus to virus. With respect to this, all viruses are divided into 7 groups by Dr. David Baltimore in 1971. He got Nobel Prize with Renato Dalbecco, Howard Martin Temin in 1975 for their work on interaction between tumor viruses and the genetic material of the cell. Next step is assembly. It involves the collection of all components necessary for formation of virion. It takes place at a particular site in the cell. For example, in pox viruses, assembly occurs in the cytoplasm. In adenoviruses, it occurs in nucleus. And the sixth step is maturation. In maturation, it is the stage of life cycle at which the virus becomes infectious. It involves the structural change in virus particles, and for some viruses, maturation occurs only after release of virus particles from the cell. Now the next step is release. Newly formed viruses are released to outside of the cell either by lysis as in bacteriophage or by budding as seen in retroviruses. Generally, non-enveloped viruses release by cell lysis which results in the death of host cell. Release of virus by budding may or may not kill the cell. This is the slide shown for release, which may occur by lysis or budding. Lysis occurs and the virus particles, newly formed viruses, are released outside the cell, and again by budding the virus particles are also released in the form of virions. Now, if we summarize all the steps in replication cycle, the first one is the attachment. As in influenza virus becomes attached to a target epithelial cell. Secondly, penetration occurs. The cell engulfs the virus by endocytosis. Thirdly, uncoating occurs. The viral contents are released by uncoating. And the fourth step, biosynthesis occurs. Viral RNA enters the nucleus where it is replicated by the viral RNA polymerase. Fifth stage is assembly. New fudge particles are assembled here, and the last stage, that is release. New viral particles are made and released into the extracellular fluid. The cell, which is not killed in the process, continues to make the new viruses. We have come to the end of this presentation, and we can conclude that, in general terms, virus replication involves three broad stages carried out by all types of viruses. These are the initiation of infection, the replication, and the expression of the genome, and finally, release of mature virions from the infected cell. At a detailed level, there are many differences in the replication process of different viruses, which are imposed by the biology of the host cell and the nature of the virus genome. Now, thank you for listening, and you can send me feedback to my email rosheba1gmail.com. Thank you again.